on for the sake of time also, and I'm happy to introduce Dr. James Allan from the Newcastle University, and he will present on genetic susceptibility to AML. And so please, Jim, we are seeing, I think, okay, fine, thank you.
Excellent. Hello, everybody. I hope you can hear me okay.
Yes.
Excellent, good. So I'd like to thank Maria Teresa, Richard and Donatello for inviting me to speak at the meeting today. It really is a huge pleasure to join the meeting, and I'm really very sorry that I can't be with you in person. I had actually intended to travel to Rome, but as you can probably hear in my voice, illness struck at earlier in the week that's prevented me from traveling. So hopefully I can get through the talk without coughing too much. So I've been asked to speak about genetic susceptibility to AML, and I'll include a little bit at the end on treatment-related disease as well, some of the work that we're planning as we go forward. So I thought it was useful, would be useful just to have a brief discussion around what we mean when we talk about genetic epidemiology and genetic risk, and particularly in the context of both constitutional genetic variants and somatic variants. So this figure, which I've plotted, sort of stylizes what we think about the relative contributions to disease risk from both constitutional variants shown in blue and somatic variants shown in red through life course as disease develops. So at the point of birth on the left, you can see that we have very few somatic mutations in our cells, and our risk of most complex human diseases, including AML, is defined largely by our constitutional genetic profile. Of course, as we age, unfortunately, we acquire somatic mutations in our cells, and these begin to dominate in terms of disease progression and sort of overtake the process essentially, and define disease development and disease progression from that point on. So I'm going to talk a little bit about some of the work that we've been doing in the last few years, looking at genetic risk, constitutional genetic risk of AML. We know a lot already actually about germline constitutional risk of AML from familial studies. So these are studies looking at families that have or are defined by usually single variants in genes that confer risk of AML with extremely high penetrance. Now you can see the list of some of the genes illustrated in the bottom left on this slide. Let me just pull my pointer up. You can see, for example, here the number of families that have been characterized with constitutional RUNX1 variants, CBP alpha variants, GATA2 variants, etc., etc. And what you'll notice is that a lot of these genes that carry highly penetrant risk variants are actually many of the same genes that we see somatically mutated in AML, many of which we've actually already heard about today during this Congress. Now, thankfully, these familial conditions are actually extremely rare and actually contribute a very, very small, make a very small contribution to population burden of AML risk. And this is, of course, because these variants are incredibly rare and carried by very, very few people. More recently, we as a community have discovered constitutional variants in an additional gene that confers risk of AML, and that's the POT1 gene, the protection of telomeres gene that's located on chromosome 7. Now, what's quite interesting about this particular gene is that not only is it somatically mutated in AML, but we also see constitutional risk variants in this gene that confer risk of both myeloid disease, AML, and also lymphoid disease, and particularly chronic lymphocytic leukemia. And you can see on the right of my slide here a family that my group are currently working on in collaboration with Friedrich Stolzel, who's recently relocated from Dresden to Kiel in Germany. So we're looking at this family who had high or have high penetrance hematological malignancy, and include a mother you can see at the top here, who was diagnosed with cervical carcinoma and CLL. And she had two sons, both of whom were diagnosed with AML, one at 16 and one at 36 years. And we very recently discovered a truncating frameshift inducing nonsense mutation in this family that's carried by the mother and by the youngest son. We've yet to identify the variant in the elder son, primarily because we're having a few technical issues because he's been transplanted. But we think this is another example of a highly penetrant, super rare familial POT1 variant that's predisposing to AML and also in this family to CLL as well. So in contrast to these cases of familial disease, we think actually the vast majority of AML cases, the risk is defined by what's called a polygenic model, whereby individuals inherit a multitude of extremely low penetrance risk variants. And typically these would tend to be very, very common variants in the population. When co-inherited in the same individual, accumulate risk to drive the patient towards developing AML. So what does a polygenic disease look like? I've put an example up here from some of the work that we've done over the last 15 years looking at risk of CLL that we now know is a classic human polygenic disease. Working with collaborators through an international consortium that was led by Richard Hilston at the Institute of Cancer Research, we've been able to identify over 45 risk loci for CLL, and these cumulatively account for about 25% of the heritable risk of CLL in humans. And I've included the data here. You can see the latest iteration of this project, which was published in 2017. We had over 6,000 cases of CLL and almost 20,000 controls. And I've included this plot here. This Manhattan plot you can see shows the risk loci that we have identified carry risk variants for CLL. So just for those who aren't used to looking at Manhattan plots, this essentially shows the human genome sort of arranged via chromosome along the bottom axis here. And then along this axis here, we've got the p-value for association with risk of developing CLL. And the plot includes essentially about 7 million constitutional genetic variants in the human genome. And what we're looking for are significant associations with these variants with risk of developing CLL. And these are illustrated by these peaks. And when we do a genome-wide association study, there's a very high risk of generating false positives due to the significantly high number of tests that are performed. So we apply an extremely stringent threshold for significance, and that threshold is set here at 5 × 10⁻⁸ for the p-value, which is illustrated by this red dotted line that you can see along this figure. So you can see all the risk loci for CLL, over 45 of them now characterized. And if we look at, we can sort of count the number of risk alleles carried by any individual and calculate their risk of developing CLL. And what you can see is that with the accumulating number of risk variants in any given individual, the risk of CLL actually goes up quite dramatically. And for example, in patients with 8 or more of these risk variants, and remember, these are very common variants in the population. And in fact, many of us in the audience will have many of these variants. You can see here that the risk, the relative risk odds ratio goes up to over 12. So this is a classic polygenic disease and very typical of almost every complex disease that we see in humans. So what about AML? Well, we started to look at AML about 7 or 8 years ago. And we knew it was going to be very important to establish a consortium to do this. So that's what we set about doing. And I've illustrated the contributors to this consortium on this figure here, including our good friends led by Maria Teresa in Rome, all contributed data and cases to this study. And the project was led in collaboration with Friedrich Stolzel in Kiel and also Ken O'Neill in New York, who also contributed some data. So This just illustrates the pipeline for the data analysis that we use. I'm not going to go through this in detail because it's pretty standard for genome-wide association studies. But what I will say is that we arrange the data into four independent genome-wide association studies, largely based on geography. Of course, it's very important that we group the cases by geography because of slight differences in genetic variation across populations. So we've got four genome-wide association studies, each with about, just over 1,000 cases included in each study. We very carefully cleaned the data after we genotyped them using high-density SNP arrays. Each of these arrays types individuals for sort of between 300,000 and 500,000 constitutional genetic variants. So we very carefully cleaned the data to remove poor quality samples and also poor quality SNPs. We end up with a nice clean data set at the end. I should also point out, actually, as you can see on the right, that we were very careful to exclude non-Europeans from the study. So the study is based exclusively on individuals of Northern European ancestry that you can see here. So our cases are shown in red, our controls are shown in blue here that map right over the top of the European 1,000 genomes population. You see our four GWAS studies here at the end, each with somewhere between sort of 300 and 400,000 constitutional genetic variants that we've typed, high quality data. We've then, using the LD structure across the human genome, we've been able to impute or infer non-genotyped variants to give us a total data set of almost 7 million genetic variants that we can then investigate for an association with AML. So at the end of our cleaning process, we've ended up with data from just over 4,000 AML cases and over 10,000 controls. And we've essentially quite simply looked for genetic variants, constitutional genetic variants that associate significantly with risk of AML. So this is what the data looks like. This is an analysis including all AML cases and controls, so all 4,000 cases. This is the Manhattan plot that we got from that analysis. And what you can see is, in fact, that we only have one significant association, and that's this signal here. This red line is the threshold for significance, p-value 5 × 10⁻⁸. You can see that we've got this one significant association, this peak on chromosome 11, 11q13, that maps right over the top of this gene called KMT5B. If you look on the right, you can see the regional plot for this particular region on chromosome 11. You can see the gene here, KMT5B, and the association signal. And again, these are all individual SNPs in the genome, and the different colors here illustrate the different extent of linkage disequilibrium between these SNPs. And you can see the sentinel SNP, the lead SNP, i.e. the most significant SNP for association with AML, is represented by this purple triangle, purple diamond at the top. Now, the other thing to point out is that when we look at the data across all four genome-wide association studies, we get a very nice consistent association with an odds ratio of around 1.2 or between 1.1 and 1.2 for all four studies. Very consistent with no evidence of heterogeneity at all across all four studies, and a very significant association. So the other thing to point out actually is that this particular risk variant is incredibly common. So you can see here the risk allele frequency in Northern Europeans is almost 50%, 0.49. So it's very common. So the vast majority of the people at Congress will actually carry at least one copy of this risk variant for AML. But the good news is, on the other side of the coin, is that the relative risk conferred by this variant is actually very low. So you can see here the odds ratio is in fact only 1.17. So this is one of the reasons why we needed a huge number of cases and controls to find this variant, that the relative risk is actually very low. So I've mentioned the gene to which this variant localizes, this KMT5B, which encodes a lysine methyltransferase. This gene is highly mutated in human cancers, and it is mutated in AML, but it's not very highly mutated. And in particular, it's been reported to be mutated as MDS transforms to AML. Seems to be occur certainly more commonly in that scenario. And this particular methyltransferase is obviously part of a large family of methyltransferases that have history in AML, if you like, and include, for example, KMT2A or MLL, which of course is very commonly mutated in AML. It's worth also pointing out that we did an expression quantitative trait locus analysis on this particular variant to see which genes in the local vicinity this particular variant was associated with expression level in whole blood. And I've listed the genes that are significantly associated with this particular variant in terms of expression. So there's in fact 12 genes within 500 kilobases of this particular signal that may be causative for this particular signal. Interestingly, KMT5B is not one of those EQTL associations, which is really quite interesting. So we still have some work to do to really properly understand what's driving the risk of what's functional at this particular locus. Now, we also did several subgroup analyses, and I'll tell you about one of these. We did an analysis looking exclusively at cytogenetically normal AML. So this obviously is AML that doesn't have any overt cytogenetically visible alterations, translocations, monosomies, trisomies, etc., or large gross deletions or gains. And interestingly, we got a new signal appeared at chromosome 6 at the HLA locus, the human leukocyte antigen locus, a very, very significant association with an odds ratio of 1.72. And again, the risk variant here is extremely common. 0.89, so carried, representing almost 90% of all alleles at this particular locus. And again, you can see the regional plot, the sort of zoomed in plot showing the signal right over the HLA locus. And I've also included again the forest plot showing the data across all 4 of the GWAS studies that we conducted. And again, you can see very nice consistency of association. There is a little bit of heterogeneity across the studies, but it's not significant. So we're confident this is a genuine association. So this is really quite interesting because HLA, of course, is a really critical region in terms of defining risk for human cancer. So this region, of course, is located on chromosome 6 and contains many hundreds of genes that encode proteins that function in the immune system, the adaptive immune system. And particularly the HLA components are involved in presenting peptides from foreign substances, viruses, bacteria, etc., to effector cells of the immune system. We don't think that particular mechanism is operating here in AML, but we do think, in fact, that the HLA system, the immune system, is operating to identify neoantigens presented by mutant AML cells. So, in fact, we did a little bit of investigation to look at this a little bit closer. So we stratified our cytogenetically normal cases by 2 commonly mutated genes in AML, which have already been discussed at this Congress quite a bit, nucleophosmin and FLT3. And if we rerun the analysis, stratifying by whether cases were positive for mutation or negative for mutation at either of these particular loci, we see that the association with risk for developing AML by this particular HLA variant is in fact much stronger in those cases that have mutant disease. So this is really quite strong evidence that the immune system in this case is operating to recognize nucleophosmin and FLT3 mutant, presumably pre-leukemic AML cell clones. And of course, we've known for a long time that many of these mutations can function as neoantigens for immune system recognition, including particularly nucleophosmin here. And I've included this table from a review published a few years ago that reports on some of the neoantigens that we know are generated somatically by malignant cells in the hematopoietic system, including AML shown at the top. So I mentioned nucleophosmin and FLT3. but also you'll recognize obviously the IDH mutations and also some of the other fusions, PML-RARA, for example, the T1517 fusion, oncoprotein is a recognized neoantigen for immune system recognition. So this just really illustrates the importance of the immune system as a mechanism for defining risk of developing AML. So I wanted to give you a little update. The data I've just talked about, we've already published. I wanted to give you a little bit of update on where we are with this study. We've been able to add a little bit of additional data very recently that we've acquired from the Finnish Hematology Biobank. We've acquired almost 350 additional AML cases from this biobank in Finland, along with a number of controls from the Finnish population. And we've been able to add those into the study. And I just want to give you a little bit of a teaser, really, that we've got some additional signals coming up in our analysis, and including this one, for example, on chromosome 2. So this is data based on now almost 4,400 AML cases and over 11,500 healthy controls. And you can see there's a signal starting to appear here in chromosome 2. And the eagle-eyed amongst you will notice that this localizes very close to the DNMT3A locus on chromosome 2. So this DNA methyltransferase that you will all recognize as being highly mutated somatically in AML. So this signal is yet to reach genome-wide significance, but we're confident with additional data that this will probably reach that threshold and we can report it. And you can see here the forest plot showing very nice consistency of association of that DNA methyltransferase signal on chromosome 2. So I'd like to just put in a little advert, really. If anybody has any data, SNP data from AML cases or indeed controls who would like to take part in the study, please do get in touch. We'd be very happy to add your data into the study. Okay, so we can go back to our sort of stylized model, and of course we can start to fill in now some of the genes that we know are operating. And as I mentioned earlier, we've already discussed many of the somatic mutations that we know characterize AML. P53, for example, we've heard a lot about. And also now we can start to add in some of the more common low penetrance constitutional variants that we know are operating in AML risk, KMT5B and HLA, and potentially DNMT3A as well, in addition to all of those high penetrance variants that I mentioned earlier in genes like GATA2 and RUNX1, for example. So I just want to excuse me. I just want to spend the last few slides discussing treatment-induced leukemia. And of course, this is a slightly different scenario because, of course, risk here, as we've heard about already, is driven very heavily by high dose exposure to either chemotherapy and or radiotherapy. And it's these exposures that we're confident drive somatic mutation in target cells that can ultimately lead to treatment-induced leukemias. Now, it's reasonable, I think, to presume that constitutional genetic variants could in fact modify these exposures and also critically modify how cells, target cells, in this case normal hematopoietic stem cells, potentially respond to those exposures, including variants in, for example, DNA repair pathways and cell death signaling pathways. So I think that's reasonable to assume. And in fact, we've got some evidence that this is in fact the case. Which I'll come on to in a minute, but this sort of suggests a hypothesis really that because of this very strong exposure that's a major driver for risk of treatment-related leukemia, that we may suppose in fact that risk of TAML or TMN is in fact driven by a small number of gene exposure interactions that are very specific to the particular exposure that's driving this risk. So as I mentioned, we do have some evidence that this is operating, and my group has worked quite a bit on TAML following exposure to methylating agents, and Maria Teresa alluded to this right at the beginning of the session. So I've listed some of the methylating agents here that we know are leukemogenic in humans, procarbazine and dacarbazine particularly. And these are used historically and in fact still currently used in some scenarios to treat patients with Hodgkin lymphoma. And they're established human leukemogens. But they're also extremely effective chemotherapy agents. And they're effective chemotherapy agents because they specifically induce this lesion in DNA, the O6-methylguanine lesion that you can see here. This is a particularly important lesion because the lesion is added to this oxygen atom, which is normally one of the hydrogen bonding atoms in DNA. And the addition of a methyl group here prevents this atom from hydrogen bonding. And in fact, what we see then during replication is misincorporation of an incorrect T base opposite this G. Normally, we'd obviously expect a GC base pair, but we get misincorporation of a T base. Now, this lesion is highly cytotoxic. It drives cells towards apoptosis, and this is effectively why these methylating agents are very good chemotherapy agents. But this process, cell death, is almost entirely dependent on having a functional DNA mismatch repair system in the cell. So where we have competent mismatch repair, and this is a little bit paradoxical because it's a DNA repair system, we would expect this repair system to in fact repair the lesion, but it can't. It attempts to repair the lesion but fails, drives the cell towards apoptosis. In this context, we have low risk of TAML because in fact the cells have died. In contrast, where we have loss of mismatch repair, or we have in fact downregulation of mismatch repair, we see that these cells survive, and in fact they have a very high risk of mutagenesis because this mispair gets fixed to a mutation. And it's this scenario that we think drives risk of TAML. So we might predict, therefore, that constitutional genetic variants in DNA mismatch repair genes do in fact predispose to risk of developing TAML after methylating chemotherapy. And that's in fact exactly what we see. So my group published a paper quite a number of years ago now reporting on a genetic variant in the core promoter of the mutal homolog 1 gene, the MLH1 gene, which is one of the major mismatch repair genes. So this variant sits in the core promoter, and in fact, there's been some very nice functional studies done demonstrating that this variant can affect the propensity of the MLH1 promoter to become methylated, which in turn affects expression of transcript and protein from the MLH1 gene. And so in fact, we reported that this particular variant is defined by this reference sequence number here. This particular variant is a risk factor for developing TAML after Hodgkin's lymphoma, but critically, only in patients who've been treated with methylating chemotherapy. We do not see this variant as a risk factor in other contexts, only in those patients who've been treated with methylating chemotherapy. This particular gene exposure interaction subsequently been validated and demonstrated to be operating in colon cancer, and particularly one type of colon cancer, and that's colon cancer operating or developing in the right side of the colon, the right side to the splenic flexure. This type of colon cancer we know is driven primarily by exposure to endogenously formed methylating agents in the gut. And this very same variant that predisposes to risk of TAML in Hodgkin's lymphoma patients treated with methylating agents is also a risk factor for right-sided colon cancer that has a strong etiological driver in terms of exposure to methylating agents formed in the gut. And this was a very nice meta-analysis done by Richard Hulston on this particular variant that included some of our data actually. So, we're currently working on the basis that TAML or TMN may in fact be essentially a monogenic disease in some contexts where risk is defined by a relatively small number of high penetrance variants interacting with these very specific exposures. And in fact, there's been several studies that have really demonstrated this principle, including some from our friends in Rome and also several from my group, where we've demonstrated really nice gene exposure interactions in determining risk of developing TAML. However, these studies have largely focused on candidate gene studies, and have typically focused on fairly common variants in the human population. But what we'd like to do now going forward is really shift our focus and leverage our genome-wide data that we've generated on our AML case set, which includes several hundred cases with TAML. We'd now like to shift focus really and start to look for potentially these more highly penetrant variants that are very likely to be lower frequency. And a lot of these variants actually won't be captured by the SNP arrays that we've used in our study, and we will likely have to sequence, use NGS sequencing technologies to find these variants. So again, I'll just put out a little advert really. If anybody has any NGS data on any TAML or TMN cases that they would like to be included in the study, then please get in touch. And just very finally, a colleague of mine at Newcastle has asked me to also put out an advert. I'm presenting lots of adverts now, which I think is great, actually. And I hope, I'm sure one of the purposes of this particular Congress. So this is a study that's been done by a colleague of mine at Newcastle called Gordon Strathdee. He's very interested in the effects, long-term effects of anti-cancer chemotherapy on non-target cells in the human body, and particularly non-target hematopoietic cells in the bone marrow and blood. And he's just published a very nice study in the British Journal of Cancer demonstrating that in long-term survivors of childhood cancer treated with chemotherapy, these patients have established chemotherapy-induced changes in their methylation profiles in their blood cells. And he's very interested in looking to see whether these epigenetic changes can in fact drive risk of second cancers, including TAML, TMDS, or in fact drive accelerated aging. So if anybody has any data or any interest in being involved in this study, please do get in touch with Gordon. I put his email there, or in fact, just let me know and I can put you in touch with Gordon. So I'd like to thank you for listening and inviting me to the Congress, and I'll finish there and just acknowledge the many tens of people who contributed data and samples to these projects, and also the funders who supported our work. So thank you very much. I'll be very happy to take any questions.
So thank you, Jim, and also thank you for giving the opportunity to many of us maybe to participate to the trial. Maybe you could send us some details on the data that you need, the samples, so that we can share with our collaborators and speakers coming to this meeting, and they will get in touch with you on the several projects ongoing at the center.
Yes, absolutely. I'd be very happy to do that, Marie-Therese. Yes, thank you very much.
Okay. Are there questions? Please, Anna. Microphone is coming, Jim.
Excellent.
Thank you for your nice talk. Could be a naive question. I wonder whether in the polygenic risk score studies you can validate in some way your hypothesis. So would it be possible to make like CRISPR-Cas9 variants in different genes and understand what are mechanisms underlying this interaction?
Yeah, absolutely. I think that's going to be required actually to demonstrate functionality of some of these variants. I mean, the KMT5B gene was quite interesting because when we looked at it after we discovered it as a risk locus for AML, although we found it was highly mutated in human cancer, its mutation frequency in AML is actually very low, which surprised us. You recall from some of the early slides in my talk that many of the familial risk variants for high penetrance AML are in fact commonly mutated in de novo disease. So we were expecting to find these low frequency polygenic variants to also localize to frequently somatic variants. And I think the DNMT3A variant that we found, potentially found, actually, we do need to validate that with some more data, but that certainly fits the model of falling in a gene that we know is somatically mutated with high frequency in AML. So for genes like that, I don't think we need to do functional studies because they've already been done. But for genes like KMT5B, I think you're absolutely right. We do need to do a lot more work, CRISPR-Cas9, to engineer those variants and really work out what the functionality is. I have to say that we're not absolutely convinced that that particular signal on chromosome 11 is driven by that methyltransferase. It is a good candidate, but we remain very open-minded about other potential risk genes on that particular locus in that region that might potentially be operating and driving AML risk. So we do remain open-minded about it.
Thank you, Constance.
Thank you, James. So I just have a question with regard to the DNMT3A mutation you described in your study. Is this the typical hotspot mutation we do see in AML, or is this outside the hotspot?
Yeah, it's actually outside of the gene. So one of the interesting things about constitutional risk variants for, in fact, all human diseases is that the vast majority of them localize to non-coding sequence. And in fact, we know that they typically don't therefore affect coding sequence and amino residue sequence. What they do affect, however, is expression, transcription levels and expression. So we haven't had a particularly close look at this particular variant to see whether it affects DNMT3A expression, but it remains a possibility that that's what's operating. But typically that is in fact what we see for most constitutional disease risk variants across all human diseases, in fact, that they do affect transcription. And in fact, those differences are often quite subtle. They're really quite small differences in transcriptional expression associated with these risk variants. So yeah, I hope that answers your question.
Thank you.
One last question.
Thank you very much for your nice talk. I have a comment about your hypothesis regarding to methylation of the methylation process. There is some data in the literature that therapy-related myeloid neoplasms has when they are treated with hypomethylating agents, they have better, in terms of CR, they have better outcomes than de novo AML. This, I think, this is an evidence for your hypothesis related to methylation. This is one. And my question was about the Dr. Dohnar's question. Well, I'm a clinician, I'm a hematologist, and I'm wondering about when we are talking about the genetic mutation, is it the same for all the individuals, for individual patients, or are we generalizing the name as mutation? For example, if we say that there's P53 mutation, is it as the same place as the other individuals?
I'm not sure I quite understand your question. So are you talking about high penetrance constitutional variants?
Not actually, for example, if the mutation is in the cytosine and all other individuals has the same mutation at that point, is it do all the patients have the same mutation at the same point? When we say that there's, for example, FLT3 mutation, if you say.
So in terms of the familial disease, what we see is typically the genes are recurrent. So GATA2, RUNX1, P53, etc. But often the specific mutation is different in different families. We do see some mutations, some particular base positions recurrently mutated, but there are numerous different variants, but the common thread is they occur in the same genes, and it's a relatively small number of genes. For those, certainly for those familial high penetrance susceptibility variants. For the more common variants, because of the way the experiment is structured, we're sort of predisposed to only find, you know, the same variant. We're looking at the same variant. So, for example, the KMT5B variant that we found as a risk factor for AML, that's exactly the same variant that we see in all these individuals. And it's a very common variant. Many of us, you know, taking part in this congress will carry that variant. So it's very common. Now, that doesn't mean that there may, you know, there may not be other variants in that gene that also predispose to AML. That's entirely possible. And in fact, when we look at the somatic mutations in KMT5B, we see them spread throughout the gene. Because, of course, there's lots of different positions in every gene that if you mutate them could affect function of the protein. But as I mentioned earlier, that in terms of constitutional variants, certainly the ones that have been discovered through genome-wide association studies and the low penetrance variants, the ones that only increase risk a little bit, most of those actually affect transcription rather than affecting sequence directly.
Thank you.
Okay, so thank you, Jim.